We now have reached the highlight of our evening, something that we all have been eagerly waiting for, the speaker's session. We have among ourselves our alumnus of St. Xavier's College, Dr. Preetha Ghosh, who is also a student of Arup Kumar Mitra sir, an ex-student of the Department of Microbiology of St. Xavier's College, Autonomous, Kolkata. Dr. Preetha Ghosh did a schooling from Gokhale Memorial Girls' School. She did her BSc in microbiology from St. Xavier's College, Autonomous, Kolkata, followed by her MSc in biotechnology from Calcutta University. Dr. Ghosh has done her doctoral studies at National Center for Biological Studies, NCBS, TIFR, and a postdoctoral research from NCBS, then IIMCB in Warsaw, Poland. Dr. Ghosh will be here to enlighten us on recent research on SARS-CoV-2 genome, which is the cause of present pandemic situation. Following this, she will be guiding us on various career options in academic and industrial research sectors. I request ma'am to address our audience. Thank you.
Thank you, Sonalika. Good afternoon, Father Principal. Good afternoon, Dr. Mitra. I'll share my screen. Just let me know if my screen is visible to you. Is my screen visible?
Yes, ma'am, it is.
Yeah. So I will mostly talk through how my journey has been, and then most of the time allotted to me, I'll like to discuss with all of you. I mean, this has been the tradition in St. Xavier's College from the beginning. Arup sir has been very enthusiastic about us interacting with our seniors, and I can assure you that helps a lot. That has helped me a lot in my journey. And I have been in touch with all my seniors, and they have kept on guiding me whenever I have reached out to them. So I have given my contacts here. Feel free to connect with me on LinkedIn or Twitter, or reach out to me on my Gmail. So I'll start. I started my journey in biology right here in this department. You can see the red arrow mark. That's where we started. So when Frederick's batch, our immediate seniors, and we started, we were the first autonomous batch of St. Xavier's College, and Frederick's batch was the first batch of microbiology. So yes, we worked along with our teachers to set up this department. So that's why it's very close to our hearts still. And I'm sure Arup sir will agree. So I got a lot from this department. Even now, I'm in touch with all of my teachers. I reach out to them for suggestions, and they have never turned me back. This is where I got the platform. I learned how to look ahead and how to just not remain confined to books, but how to read articles, how to make presentations. But even maybe 15 years down the line, when I talk here, there is a lump in my throat always. But yeah, I will take you through how it has been after that. So post my bachelor's in microbiology, I moved to the University of Calcutta for my master's in biochemistry. This departmental corridor might be known to many of you. We have a lot of alumnus of this department also in St. Xavier's. I did my master's from here in biochemistry, and then I moved on to do my doctoral studies and my first postdoctoral research on this beautiful campus in Bangalore, the National Center for Biological Sciences, TIFR's biology campus. And this is where I got introduced to RNA binding proteins when I looked at sequence and structures of RNA binding proteins. And this is my mentor, Professor Sogamini, under whom I did my doctoral and first postdoctoral research and my lab, all thanks to them that I got a quickish PhD and I could do a quick postdoc also. So I will tell you why specifically RNA binding proteins. DNA binding proteins like transcription factors and replication factors, they have been looked at a lot. people. But what has not been looked at by people are that much, at least at that point, 2012, when I started my PhD, was RNA binding proteins. They are gradually gaining limelight, but it was important for us because they meet very diverse functions of this cell, especially because they are proteins like the ribosome and the spliceosome, which are the nerve house of the cell. Um, they regulate co-transcriptional and post-transcriptional gene expression, which is a very important factor for the functioning of the cell, and not only the normal functioning, but whatever irregularities happens, like cancers and all, also happen because of malfunctioning of gene expression. And like I said, they are the structural components of the ribosome and the spliceosome, which are the biggest machineries of the molecular machineries of the cell. So they are composed of multiple copies of RNA binding proteins along with their binding RNAs. And then I looked at RNA sequence and structure guided recognition. So there is a reason why I'm telling you all this about RNA binding proteins. Some of you might be able to make a connection based on the introduction that was already given. So during my career, I got trained in microbiology first, and then I looked at RNA binding proteins, and I kept on looking at RNA binding proteins. And during the pandemic, maybe it all fell into place when we were looking at a virus with an RNA genome. So after my first PhD and first postdoc, I moved out of India. I went to this not so known place, the Warsaw, the capital of Poland. Uh, I chose more the lab, where I looked at the large scale sequence analysis and structural modeling of complex structures of RNA. As most of you know, RNA takes much more complex structures as compared to DNA, and their interactions with proteins, like I mentioned in the previous slide, and ligands, like mostly drugs, are what defines their multitude, their breadth of functions, I would say. So this is what my postdoc was destined to look at. This is my postdoc mentor, the science advisor of the European Union, Professor Janusz Bujnicki, and my lab in Warsaw. I learned a lot here. I learned how to collaborate with people from very different dimensions of biology. I learned how to collaborate and work with people coming from diverse backgrounds. So in this lab, there was nobody who had overlapping backgrounds. There were computer scientists, mathematicians, and this is what biology currently is all about, interdisciplinary science. Because you can't just say, I'll stick to biology, I'll not do physics, I don't know much of chemistry. I mean, all the disciplines of science now go hand in hand. And this is what I learned mostly from this postdoc. I was happily looking at RNA binding proteins and their RNA again, which was always my interest. I was looking at protein families that have been conserved from Drosophila, the fly, to C. elegans, to worm, and to human. And we were getting like very nice insights on the evolutions of proteins until this happened. So we landed onto the pandemic, and the entire scientific community had to just set aside what they were doing, and we had to save the world, sort of, like you see, like Sir also said, only science can pitch in and solve what is going on now. So that's when I tried to put in my knowledge of microbiology, structural bioinformatics, sequence bioinformatics, looking at RNA, and maybe the pandemic positively affected my research. So what we did as a part of this study is, when the entire world was running against the virus and people were running behind vaccines, what we and our team did is we sequenced the entire genome of the wild type coronavirus, And then nucleotide by nucleotide, the entire 30,000 nucleotide, we looked at their secondary structure. Now, why was it needed? So only if you need to know curb the replication of the virus, you need to know how the virus looks, what are its strengths, what are its weaknesses, where are the pockets where drugs can go and sit. For vaccines, it was a parallel line of treatment already going on. But for drugs, we didn't have much idea till this work of ours, where we first sequenced and solved nucleotide by nucleotide the entire structure of the coronavirus genome. And the experimentally determined structure were then made used to make secondary structure predictions, and then mapped onto the genome to identify like structured domains or pockets in the genome where a drug can go and bind. And I still remember sleepless nights of going nucleotide by nucleotide and looking at the entire 30,000 nucleotide, but yes, all for the good now. And this is a bigger picture of what we achieved. So you see different RNAs here. These are different parts of the coronavirus genome. We see these are pockets here. You see grooves here where a drug can come and bind and prevent the folding of this RNA, in turn leading to blockage of the replication of the virus, leading to remission of the infection. So, what we did as structural bioinformaticians with knowledge of microbiology was we modeled the domains based on conformational search. So, this is a way by which we simulate biomolecules, look at what all possible structures these molecules can take, and look at their chemical feasibility, because we also need to, the chemistry plays a huge role here. We need to see what is feasible in terms of allowed structure and what are disallowed structures. And then after we had modeled, we redetermined the secondary structure to be sure that these generated models were on the right track. Uh, in the process, I ended up collaborating with a lot of people from all over the world. It was a huge learning experience. They had very different kinds of expertise, starting from my days of PhD, as well as during my second postdoc. Um, they were all people with different expertise, and I had to learn very fast. And in this respect, I would say please always keep your mind open, especially in this age of interdisciplinary science. You need to accept change. You need to move out of your comfort zone to welcome newer techniques, which you might need to at times learn overnight to start implementing them. Of course, there'll be experts and people to guide you, but do accept change and delve into not so known areas. And that's how I'll summarize my skill sets. I do some bit of sequence bioinformatics, which I have done for SARS-CoV-2, a lot of structural bioinformatics again for both protein and RNA in SARS-CoV-2, like I said. Next generation sequencing techniques. So this is like the future of biology. So whatever we know about all the mutant strains that are coming up are all courtesy next generation sequencing. And large scale data analysis. This is something I am doing currently in my company. So I've joined this company in Bangalore called Entrance, which works hand in hand with the hospital Mayo Clinic in the US. And we try to use artificial intelligence based methods to make sense out of biological data. So mostly I'm working on more clinical patient records now of COVID-19 patients, all what have been prescribed by doctors in their notes, or what have been given by, what medications have been given, how long have they lived after being given a medication, what kind of comorbidities drive their survival rate, and so on. Yes, I would like to end by thanking all of you for your kind attention. Thank you, sir. Thank you, Father Principal. You see Arup sir here. And this is me here with my entire batch, all my teachers here who have taught me and still advise me whenever I'm lagging behind, and Nihil Ultra. So I would like to take questions maybe.
Yes, ma'am. Please give us a minute so that we can get the questions
Yeah,
and
yeah,
address
sure.
it.
With your permission,
Yes, sir.
I am leaving the meeting. I
Okay,
enjoyed
sir.
your talk. Please
Thank you.
interact with the students,
Sure.
motivate them so that they can also have a good career ahead of.
Thank you, sir. Thank you
Okay.
so much.
Thank
Thank
you.
you.
Thank you.
Thank you, sir.
Thank you, ma'am, for your patience. So we have got a few questions from our students. So the first one being, how was your overall experience in pursuing a doctoral and postdoctoral course?
Okay, so doctoral, so I'll be very frank here. So after I finished my bachelor's and master's in experimental biology, as a mainstream, I joined experimental biology as a PhD, but that was not my cup of tea. So I realized within a year that's not something I was liking doing. That's when I switched to bioinformatics, which was never my expertise. But I realized that I am liking looking at structures, biochemical structures, the structures of viruses, bacteria, which I have always learned rather than looking at them experimentally. So I always like the broader picture. I was extremely lucky to have an excellent PhD mentor. So she guided me throughout, and PhD, as you know, requires a lot of patience. There'll be more failures than there'll be successes. So that way, and being in a place like NCBS where you compete with the best in the country, I had very good batchmates, colleagues who always got the best out of me, I would say. So yeah, so they always kept on motivating me. Yeah. For postdoc, it was quite different. In Europe, the work culture is very different. They are very different from how things work in India. So it was way more professional. So I think I learned a mix of both from both my 10 years.
Thank you, ma'am.
Yeah.
Ma'am, the sorry, just a second. So the next question is from another student who asks, so the drugs are targeting the virus or the receptors to which the spike binds?
Okay, so these drugs are mostly targeting the virus. So, like you are right, there are drugs which are being targeted to break the interaction between the receptor and the spike. Mostly the vaccines target spikes in a place which can hamper the binding with the receptor. But in our case, it was mostly targeted to the virus itself. Because when the drug binds, the RNA of the virus changes conformation such that the proteins that facilitate its replication cannot sit and bind and recognize and carry on with the replication. Means the virus cannot replicate anymore, leading to the clearance of the virus from the host. If
Thank
that
you, ma'am.
answers, yeah.
You have a third question. There's some problem with my video. Could the host please check once? I'm not being able to turn it on. Yeah. Thank you. So the third question is, what are the main challenges you faced while sequencing such a large genome, even though the most advanced techniques are in place?
So actually 30,000 nucleotides is not a very large genome. It's very large compared to viruses because viruses are usually never this large. The coronavirus is one of the largest family of viruses in terms of genome size. But they are not, I mean, NGS methods, I mean, the biggest genome I have worked with are plants, which are bigger than humans in size. So actually 30,000 nucleotide is not a very big size, but then I can tell you So with plants, which are bigger than human genomes, you need to have multiple copies, multiple libraries, very good quality DNA, so that you cannot sequence the entire thing in one shot. So it's broken down based on the platform that you use or the enzymes that you use. So it's sequenced in pieces, exactly like how crosswords are. So, and then you stitch them based on overlap. So, for example, if I say my name is Preetha Ghosh, I can't sequence Preetha Ghosh at one go. So PRITH maybe gets sequenced at once. Next, THAGH gets sequenced, and then OSH gets sequenced. So I have to look at where all they are overlapping to know which fragment comes after which fragment. So that is a big challenge. That's why NGS techniques are very expensive, because in order to get good coverage, to sequence the entire genome, we need to run a lot of sequencing runs. We need very good quality DNA. And that's what makes NGS very expensive. But once you get a hand of the technique, there are experts always who are doing the sequencing, though we are the ones who prepare the DNA. And the DNA quality also makes a huge difference. Mm, and also the sequencing machine. There are newer sequencing machines coming up which give very high depth of sequencing, means how many times a part of a genome has been sequenced.
Thank you, ma'am. The next question goes: is the virus really man-made, though?
No, no, and not at all. It is not at all. It comes from bats. And you'll be surprised to know that the SARS-CoV-2 is more similar to bats than they are to the previous SARS-CoV. The 2003 pandemic causing SARS-CoV is less similar to the current SARS-CoV-2 than it is to bats. So it is not at all man-made.
Thank you, ma'am, for the much confused, like
Yes,
the much confused question among many of
yes,
us.
yes.
Thank you for clarifying. The fifth question is: how much of a disadvantage will it be for us college level science students to not be able to learn lab skills here due to the pandemic? Which is a very serious question.
Yes, this is a serious question, and also this is concerning, because when you start your PhD, when I started my PhD, nobody taught me how to set up a gel, say which is a gel, or how to set up a gel. These are things which I was supposed to know courtesy my prior education. So they taught me advanced techniques as to say how to make a thicker or a thinner gel, as to how to vary the pore size of a gel when I'm loading it. So this is a serious disadvantage at the moment, for sure, because you're not getting practical lab skills. As opposed to that, something which we never had, which you guys have, is YouTube. There are loads of good tutorials where you can actually see the instruments, though you cannot handle it by yourself, but you can actually see the instruments, so that you know which is what, how to fit a gel together, or which electrode goes into which end of the gel plates, and all that you can already see. But all the people who will be starting off with their PhDs along with you will be on a similar boat. So you will be learning together, albeit the learning time will be slightly more because you have lost out some time during your bachelor's and master's. But I would say, take this time when you are not able to get done with your practicals to see more and more illustrations, more and more videos. I see more videos coming up every day of practical science who show lab techniques and all, to at least be aware of all the things that are going on, all the kind of instruments that can be used for different scientific measurements. Yes, but there will be people who will be ready to train you. Just do a bit of more background research than we had to do, so that when people are teaching you, when you start your PhDs, you are not absolutely blank. You at least have some idea what they are talking about.
Yes, thank you. Um, so another question goes. It's a career related question. It says, I love biology and I'm currently studying BSc in my chemistry honors. How can I pursue a career in biochemistry?
Okay, so half my class who were in masters were chemists. So it's not that you have to study biology in order to do biochemistry masters. So nothing like that. So that's one thing. And the second thing is, yes, the way maybe the syllabus of Calcutta University is designed, biochemistry is not 50-50 biology and chemistry. It is a bit more of biology than it is chemistry. But trust me, in the long run, you will see how important a chemist's position is, because a chemist knows a good deal of physics, a chemist knows a good deal of mathematics, a good deal of biology. So that way, I think a chemist is the best placed amongst all the people from all other disciplines. As of to get through your masters, if you want to go to something like biochemistry for your masters, use this time to understand the basics of biology. This is something I had seen with my classmates who came from chemistry bachelor's streams. Uh, because the master's syllabus of Calcutta University is massive, I must admit. So you will not have that much time during your masters to understand the basics of biology. So maybe spend this time. You spend your free time. This will take, because you're saving quite some time from traveling also to college and back these days. Spend that time to understand the basics of biology, how the cell works, what are molecules, what are proteins. These are the most important things. So even though you know the chemistry, you don't know how it is in the biological context. So learn a bit about proteins, amino acids, carbohydrates, lipid molecules, the cell in general, how it functions, and the rest all you will be at par with all your colleagues who come from biology.
Thank you, ma'am. The other one says, won't a lot of medicine need to enter the system to target this virus? What have been done to counter or reduce the side effects?
Uh, Side effects. Oh, yeah, this is so the most important thing about side effects which can be dealt with is clinical trials, as opposed to previous viruses for which vaccines and drugs have been developed. We didn't get that much time to we didn't get that much time to deal with the side effects this time. But we can assure you that all the drugs that go into clinical trials and are then marketed have a minimum of side effects. Of course, it will be different from human to human because every body is different. There'll be something like fever or nausea or vomiting, which allergic reactions which will come. So before you take a drug, always consult your doctor, because you might be having something really rare which other people don't have. You might have some allergy which is very rare and which was not taken into account while designing the drug. The composition of the drug might be not the best for you. So yes, I mean, but almost all the drugs that go into the market um, have been already screened for minimizing the side effects.
Thank you, ma'am. The next question has got several parts. It says, is it possible to reduce COVID 19 to zero? Like, is it absolutely possible to kill a virus? Is a virus living or non living? How did they originate in the first place? And is COVID 19 a newly made virus or was it present in some form for many years?
Okay, I'll go one by one. So virus, living or non-living, this is one of the, I guess, fundamental things in virology. The virus is not living outside a host cell and it's living inside a host cell. So it means a virus cannot replicate outside the human body or the host's body, in this case human. and it can replicate only when it's inside a host's body, because the virus does not have all the machineries to need which are needed for its replication and thriving. So it needs a lot of host factors like replication enzymes, a lot of host proteins to thrive there. So it does not live outside the cell, it lives only inside the cell. It is not possible to kill a virus completely. Yes, like that's the answer. Coronavirus or any other virus for that sake will never be completely gone. And that's the precise reason why existing viruses change over time, adapt to different conditions, and come back in a completely new avatar. That's what exactly has happened here. The coronaviruses are one of the largest family of animal viruses. They were never gone. They just keep coming back in different disguises, different avatars, as you say, by mutating over time in different hosts. Previously, it was in bats. From when humans started eating bats, it came to humans and changed itself to adapt to the human body. And that's why first human don't die, and it's not completely novel. Its nearest neighbors were there, they are still there, and they will be there. And that's why most of the research that is going on now is to curb the spread of a recurring pandemic. Did I miss on something, Sonalika?
No, ma'am, it was absolutely fine. Thank you. There's another question. We'll try to wrap it up as quickly as
Yeah,
possible.
no problem.
Uh, it says, did you require special permission to sequence the genome? Is this possible in college laboratories in India, like at St. Xavier's College?
No. So there are special sequencing machines. There are special sequencers, and especially in India, there are only specific labs for, especially for viruses. So in general, okay, next generation sequencing can be done only using special equipments called next generation sequencing platforms like the Illumina HiSeq or the Roche. These are special machines which are extremely expensive and either are funded by the government of India or comes from funding from private organizations, and are present only in specific institutes or labs who specifically have funds for them. My PhD institute had a sequencer, had multiple sequencers actually. That's where we did all our sequencing. I am not aware of any college per se having their own sequencer because these are extremely expensive and most colleges don't get funding for this much funding for research. Secondly, what was that you asked? Okay, so for sequencing of viruses, uh, in India, viruses can only be handled in biosafety levels 2 or higher. And NIV Pune, the National Institute of Virology Pune, they have special permissions for it. So does the Serum Institute of India. So you cannot handle human viruses or lethal viruses like this in normal laboratories. You need specific biosafety level labs for doing handling viruses, even if it's sequencing, any kind of assays, everything. So all the experiments that I showed you were done in labs which are specially equipped, biosafety level 3 labs in Europe. These were done in Netherlands, where they had special permissions from the European Union to do these sequencings.
Thank you, ma'am.
And I mean, just to add to it, I mean, in general, this pandemic is different. But at any point, if you want to sequence a viral genome, you don't need special permission every time to go to people and ask. But here, because the time was less, efforts were concentrated, every effort and time mattered. That's why there were central registries who were keeping track of which virus is being sequenced when. Yeah.
Thank you, ma'am. Uh, Another question goes, why is the using of calcium chloride in ammonium sulfate for salting of proteins not used to eradicate coronavirus?
Okay, because coronavirus is inside your cells. So you use calcium chloride and sodium carbonate to precipitate proteins that is inside test tubes and ependors, not inside the human body. So if you use calcium chloride and sodium carbonate inside your cells, not only, okay, the virus will be dead, but you will be half dead too, because it will be equally affecting the viral proteins as well as your proteins.
Thank you, ma'am. Okay, we have okay, we have another question. Are not coronaviruses sensitive to heat? Because this is what we have heard for a certain period of time.
Yeah.
But how did they come into human body via food? Because food is usually heated before eating.
Yes, so that is one thing. But then this came while a certain sect of people were eating raw meat. In cooked food, if you reach boiling point, then coronaviruses are killed. They can't thrive beyond that point. So, like spores are endospores. But coronaviruses didn't come to our body through cooked food. It came when people were eating raw meat.
Thank you, ma'am. Why bats can harbor many potential pathogens in their microbiome, but still they manage to survive this long?
Okay, so bats also die. I mean, they are also equally affected by coronaviruses. Just that, so I know a lot of my colleagues who do behavior experiments, so animal behavior, where because we cannot distinguish between which bat I had looked at yesterday, right? So they tag each animal, like bees or bats or mouse, with paints, uh, so not to positive negatively affect them, but for the sake of experiments. So if you inject coronavirus in a bat and mark that bat amongst all the other bats, so over a period of 14 to 15 days you can track that bat, and you can see they also develop symptoms and eventually die because they don't take medicines like us. So yes, so when they are infected, they do die, but all the bats who live long are the healthy ones. When so, if you track a particular bat over time, you will see how long they live.
I wish anyone was interested to do that. They might.
Quite a few people I know do that.
Yeah, yeah.
They look at bats.
So, yeah, I have a serious question with me that
Yes.
how is this virus mutating? Like, this is the triple mutant. Why is the second wave into life again?
Okay, so the thing is, uh, all the vaccines that have been designed till now, they were designed based on a certain pool of mutants. And the newer strains have some mutations against which the vaccine is not the best measure. Till newer vaccines come up, these are going to thrive. But all the viral mutations, they are spontaneous. They can you can't say when a virus mutates because mutation is a spontaneous process. It can be sped up by certain chemicals or something, but the virus does mutate, and especially in a population like India, where the gene pool is so varied as opposed to the Caucasian or the Western population. We have such a variety of gene pool, so much diversity in our genes. We are so different from each other, so, entering our bodies, the virus needs to develop newer strategies every time to thrive. So, what might be detrimental for me might not be detrimental for you. So, the virus, in order to replicate, might need newer strategies inside different humans, and that's why that aggravates their process of speed of mutation, I must say.
Thank you, ma'am. So this question is very important even from my side. It says what is the most promising vaccine in our development right now, because even after taking the vaccine we have seen that people who have completed 2 doses of virus have also gotten affected by coronavirus. I mean, after 2 doses of vaccine have also
Vaccine.
been affected by coronavirus. So what is the possible way of getting rid of this, which seems plausible now?
Yeah, that's a very grave point actually, because all the vaccines that were developed, like I said, were developed based on the existing strains. As more and more mutants come up, there are like the South African strain or the Bengal strain, Indian triple mutant as it called. There are insertions and deletions in the genome. There are changes which makes the antigen antibody interactions weaker, or maybe so weak that you can consider it negligible. So, for all the previous infections for which vaccines were developed, we already had a huge amount of sample, a huge amount of variety of the virus, which we already knew when before scientists went on to develop vaccines. In this case, it was the need of the hour. So, whatever sample we got, the scientists started making vaccines. But there are newer and newer strains coming up which were not pre planned while making the vaccines. So, the only way out is I know a few companies are now starting to market drugs. So, drugs may be a way out. It's something I also worked on, and newer vaccines, and people um, staying at home and maintaining social distance.
Thank you so much, ma'am. Recently, it's been said that the virus is turning out to be airborne. Is this true or is it just a rumor?
No, no, it's not a rumor. It's not a rumor. There is a scientific paper in the Lancet which confirms the fact that. So they have proposed, it's a big journal, very less chances that it will be a hoax. They have proposed, but we have to wait till somebody else confirms it. But this was also proposed by a group in the UK, if I remember correctly, in the while the first wave was going on in India, that these are airborne, but they did not have sufficient evidence. But the current paper in the Lancet has quite concrete evidence of it being airborne. But we have to wait and see till other people prove it actually.
Thank you, ma'am. We'll wrap up with two last career
Sure.
guidance related
Yeah,
questions.
yeah.
One says, is switching from microbiology honors to MSc in biotechnology beneficial for future interdisciplinary research?
Yes, yes, absolutely. So biotechnology, so there are two things. There's a Bachelor of Science in Biotechnology and there's a Bachelor of Technology in Biotechnology. So if you want to go into full-time research, I would suggest to move towards BSc Biotechnology more, because you're talking about doing it, or rather MSc in Biotechnology. You're talking about a master's in biotechnology, yes. So I would suggest a MSc in biotechnology rather than an MTech in biotechnology.
Actually, in St. Xavier's, we have an integrated course of
Yes,
both BTech and MTech, so it anyway.
yeah. We were the first batch of uh, our batchmates were the first of those, 2006.
The last question for the session. It says, I'm currently doing BTech in biotechnology. Can you please guide me what to do after I complete my BTech? So I think you already have answered this.
Yes, I mean, this completely depends on the person's interest. They can either go into mainstream research, like after a BTech, MTech, or a BTech and a PhD. But if you want to go into research, I would say directly go into research post your BTech. Uh, but if you want to, say, get a job and get more skills in the industry, then go for an MTech also. But that completely depends on the person's interest as to, if you want to go into mainstream research, then do a BTech, and if you want to stay in India, you can directly start a PhD in all the national institutes. But if you want to go go abroad to some place like the US, then you can also do an MS there and then start your research.
Thank you so much, ma'am, for taking out the
And
time.
one more thing, I think in this context, I should mention, which I tell people from time to time. PhD is a long-term commitment. You might, because there are so much failures as compared to success, you might become fed up and tired of it after a point. So I would suggest that if you are not absolutely sure, especially people in the current scenario who are not exposed to lab work, unless you actually do work in the lab, unless you actually encounter failures of experiments, you will not know whether you would like to do research long term, because PhD is a long-term commitment. So if you start a PhD and 3 years down the line you don't like it, then you have lost 3 years. You have some experience, but you have lost 3 years, and you exit there from there without anything. So you're still a master's. So especially people like you at the current scenario, unfortunate consequence of events, who are not getting exposed to projects and lab work like we did, I would say go for these small internships or being a junior research fellow somewhere. See whether you actually like doing research full time. Maybe you'll realize that yes, maybe it's as fascinating as it sounded from textbooks, but others might feel like no, 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 I can't deal with this every day, getting up in the morning and thinking of failures. Then I would say don't waste your time. PhD is not the end of the world. Take alternate careers. But if you like doing it, then take the traditional path, do a PhD and then decide what you like.
Thank you, ma'am, for your enlightening words. And I also
Thank you.
thank you for coming here and holding the session.
No problem, my
And
pleasure.
thank you for taking all the questions. We have a lot of questions still coming up, but because of the short of time, we'd like to wrap up now.
Sure.
Thank you, ma'am.
Thank you so much.